Today we're going to do a demonstration on how to do a packed cell volume reading and a total protein reading on whole fresh blood in an EDTA tube. So we'll go over the supplies that we have here. So the first thing we have is our little centrifuge. We're going to use that to spin down the microhematocrit tubes. We have our fresh blood sample. We have our little stat spin tray that we're going to use to put in our microhematocrit tubes. We have our clay sealant. I have some clean Kim wipes to clean up afterwards. I have some distilled water also to help clean up. I have my refractometer. And I have two different little reading charts for reading our microhematocrit tubes. Either works fine. I prefer this one, so I'm going to use this for a demonstration. Okay, so we'll get started. So in a real case scenario where you want to actually get a true diagnostic reading, you want to first get a fresh blood sample properly collected, usually from like a jugular vein. And you want to process it immediately. So assuming that we just obtained this sample, the next thing we're going to do is we're going to take our fresh collected blood, we're going to invert it 4 to 5 times. So that just means gently rocking back and forth 4 to 5 times. You never want to do this aggressively, or you never want to shake it. That can actually damage the red blood cells. So once you do that, we're going to go ahead and we're going to fill up our microhematocrit tubes. So because I have a little blood in the cap here, I'm just going to set my tube aside and I'm just going to do this here. So we're going to fill the tubes up to about that little blue mark, and that blue mark is kind of like our just over 3/4, 2/3 of the way. We're just going to fill it all the way, and we're going to seal the end with our thumb. We're going to switch sides, and then we're going to seal it with the clay. And you're going to want to put it in your microhematocrit slot here in your spinner, and you want to put it with the clay side out. So I'm putting it in slot number 1. Okay, we're going to fill the other 3 tubes the exact same way. So filling it, your tube should look right about like that. I'm not sure if you can see that, but I'll bring it up. The tube should look kind of like that. And when you place it in your spinner, you want to make sure that you're balancing it out. So I put it in slot 1 first. I'm going to go directly across, and I'm going to put it in a slot directly across from it, which is slot 7. And you want to do 4 samples at a time and obtain 4 readings. And just make sure that they all match up. Seal it, put it in our slot here. Okay, so we're going to put this one in slot 10. And we're going to put our final one across from the last one we just placed, and that's going to be in slot 4. Oops. Okay, the next thing we're going to do is we're going to put our cover on top. We're going to make sure it's nice and seated, nice and tight. So before you put it in the spinner, it should kind of look like this. It's all symmetrical. Each tube is across from another tube, so that way it all balances each other out. So the next thing we're going to do is we're going to put it in our little centrifuge here. I'm going to stick it in. And we're going to make sure it's on the correct setting. So this particular little centrifuge is made specifically for these kind of things. So it actually has a microhematocrit setting. So we're going to make sure it's on that one. Press the lid down gently, and you're going to press the go button, and it's going to do its thing. Okay, so now that our centrifuge is done spinning, the next thing we're going to do is we're going to open it, and we're going to go ahead and we're going to take our tubes out. Looks like they're all nice and spun down. And the next thing we're going to do is we're going to read it. So like I said, you can use either chart. Either is totally acceptable. I'm going to use my circular one. It's just a matter of preference. So we're going to read all 4 tubes. And the way we do this is you basically line up the bottom 0% line with the bottom of the red blood cells, so the red layer. And then you're going to line the very top line, the 100%, with the top of the plasma layer, just between the plasma and the air layer. So we're going to go ahead and I'm going to read this. Okay, so I'm getting about 52% for the first tube. And again, you want to read all of them. You want to make sure that they all match up. Just in case you made an error in the first one, you want to make sure that you're getting the correct results across the board. Okay. So again, you're going to line the top of the plasma with the 100% and then the bottom of the red blood cells with the 0%. And again, I'm getting about 52. And we're going to read the third tube. Getting about 52%. And I'm reading the last tube. And so just so I can explain what I'm doing here, I'm actually reading, basically, I just stated how to line them up, but the way you find your number is you read the top of the red blood cells, where that one lands when you line up properly. And across the board, I'm getting about 52, 53%. So I think we can safely say our packed cell volume is about 53%. So the next thing I'll do is I'll pause this video and we'll go ahead and we'll do our total protein reading as well. And you actually do this with our tubes that we have here. Okay, so the next thing we're going to do is we're going to obtain a total protein reading on the microhematocrit tube serum that we just ran. So we're going to make sure we write down our patient's packed cell volume in their chart. So it was 53% packed cell volume, PCV. So now that we have that written down, I'm going to show how to do this. So what you do is you actually physically break the tube above the buffy coat layer, and you want to break it at the plasma layer. You're going to basically take that plasma, put it onto a refractometer, and you're going to take a reading. So you can do this really gently. I just kind of do it over a little micro. There's kind of multiple ways to do it, but you just want to be very gentle. And you very gently break it with I use a slide. You're basically going to take this liquid, you're going to kind of gently tap, and you're trying to get that liquid out from the tube. Sometimes it can be a little tricky. And if you don't get enough liquid from that one, you can always go to your next tubes and use those two. And when you put this on the refractometer, you want to be very careful and make sure that you don't get any glass shards and any contaminants on it. Okay, you're then going to take the little bit of plasma here that you got. You're going to get a reading. So you're going to look in the refractometer and you're going to look at the left side the serum plasma side, and you're going to take a reading. So I got about 7.2. And then, so basically you match up the white and the blue where they come across, and I am seeing it's right about the 7.2. Yeah, that looks about right. And the last thing that you'd absolutely want to do is you'd have somebody double check your work. So in that case, it'd be a veterinarian. So you make sure you have your veterinarian reading. This is a very important reading. It does a very general, it can give a very good general assessment of the pet's health and hydration status. So you'd always want to make sure to have your mentor check this as well. And anyways, so that's how you get a total protein reading. The very last thing you're going to do is you're going to want to clean your refractometer, and that's very important. You'd only want to use distilled water and Kim wipes. These are laboratory-specific wipes made just for cleaning things like this. So you're going to just put a little drop of distilled water and clean it up. And then you would put it back in its case. I don't have its case here, but you would put it back in its case. So we'll gently set that aside. I'm going to clean up our tubes here too. I want to make sure you get out all the glass shards. Very important. Okay, and that is how you do a total protein reading on an animal.